Welcome to the fascinating world of developmental biology. Today, we're going to explore one of the most critical events in the continuation of life, fertilization. Imagine you are trying to send a very important, life-changing letter to a friend. You have two choices. You could either throw thousands of copies of the letter into a fast-moving river, hoping just one copy floats exactly to their house. Or you could hand deliver a single copy directly into their secure mailbox. In the biological world, these two strategies perfectly describe how animals ensure the survival of their species. We will discover exactly how and why nature uses both of these incredible strategies to create new life. Building on our letter analogy, let us understand what this means biologically. Fertilization itself is the magical moment when a male gamete, the sperm, fuses with a female gamete, the egg, to create a new, unique organism. This fusion is the ultimate goal, but the journey to get there can look very different depending on the species. Nature essentially uses two main delivery strategies to achieve this fusion. The river method, where gametes are released into the environment, is an analogy for external fertilization. On the other hand, the hand delivery method, which is much more targeted, represents internal fertilization. Both methods have evolved to solve the exact same problem, but they do so in completely different ways. Now that we understand the ultimate goal, let us look at the first strategy in detail. External fertilization occurs when the fusion of the sperm and egg happens completely outside the bodies of the parents. Because gametes are delicate single cells, this process typically must occur in a watery environment, like an ocean or a pond, to prevent them from drying out. In this scenario, both parents release their gametes into the open water simultaneously. This synchronized mass release is a process known as spawning. It is a chaotic and risky environment, which is why organisms that use this method must release massive numbers of eggs and sperm to ensure that at least a few successful collisions occur. Contrasting with the chaotic open water, let us examine the alternative strategy. Internal fertilization occurs when the male deposits sperm directly into the reproductive tract of the female. Here, the fusion of the sperm and egg happens safely inside the female's body. This internal environment is highly controlled and protected from the harsh outside world, meaning the gametes are not exposed to predators, temperature changes, or the risk of drying out. Because the delivery is so direct and the environment is so safe, organisms utilizing this strategy do not need to produce millions of eggs. However, this method does require more complex anatomical structures and behavioral coordination between the parents to ensure successful mating. Understanding these definitions leads us to an important question. Why does it matter which strategy an organism uses? The answer comes down to evolutionary survival and the specific environment the animal lives in. If an animal lives on dry land, releasing eggs and sperm into the air would be disastrous because the gametes would dry out and die almost instantly. Therefore, land-dwelling animals absolutely require internal fertilization to provide a moist, safe environment for the gametes to meet. Conversely, aquatic animals are surrounded by water, which acts as a natural medium for sperm to swim toward the egg. This makes external fertilization a viable and energy-efficient option for them. Without these specific adaptations, entire groups of species would simply fail to reproduce and would go extinct. This biological need for survival brings us to the mathematics of fertilization success. To truly understand the challenges of fertilization, we must look at it through the lens of probability. Whether inside or outside the body, it is fundamentally a numbers game based on collisions between sperm and eggs. Biologists use this beautiful equation to model the probability of success. The variable F represents the fraction of eggs that are successfully fertilized, ranging from 0 to 1. The constant E is the base of the natural logarithm. The variable K represents the rate constant of sperm-egg collision, measuring how easily a sperm can find an egg. S represents the concentration of sperm, and T represents the interaction time. Notice that if you increase the sperm concentration S, or the time T, the negative exponent becomes a larger negative number. This makes the exponential term approach 0, which pushes the fertilization success F closer to 1, or 100%. Before we move on, let us see this formula in action with a concrete example. Imagine a species of sea urchin that releases its gametes into a small, enclosed tide pool. We want to calculate the fraction of eggs that get fertilized. We are given a collision rate constant K of 0.01. The sperm concentration S is relatively high at 100 units, and the exposure time T is 10 seconds. We substitute these known values into our fertilization equation. Multiplying 0.01 by 100 gives us exactly 1, and multiplying that by 10 gives us 10. Because of the negative sign in the formula, our exponent becomes negative 10. The value of e to the power of negative 10 is extremely small, approximately 0.000045. Subtracting this from 1 gives us a final answer of 0.9999. Physically, this means that in the confined space of a tide pool, 99.99% of the eggs will be successfully fertilized. Now that we have successfully used the formula for an external environment, let us look at a slightly more complex example involving internal fertilization. Internal fertilization provides a highly confined and specialized environment, which drastically increases the collision rate constant K to 0.1. We want to find out exactly how much sperm, S, is needed to achieve a high fertilization rate of 99% in this efficient environment. We set F to 0.99, K to 0.1, and keep the time T at 10 seconds. Plugging these into our equation, we get 0.99 equals 1 minus e to the power of negative 0.1 times S times 10. Multiplying the rate constant by the time simplifies our exponent to simply negative 1.0 times S. This sets up the equation we need to solve to find our unknown sperm concentration. This equation we just set up leads us to an important mathematical proof of biological efficiency. We need to isolate the unknown quantity S, which is currently trapped in the exponent. Our starting point is 0.99 equals 1 minus e to the power of negative S. The first algebraic step is to isolate the exponential term by moving things across the equal sign. We do this by subtracting 1 from both sides of the equation. 0.99 minus 1 gives us negative 0.01. Now we have negative 0.01 equals negative e to the power of negative S. The negative signs on both sides cancel each other out perfectly. This leaves us with a clean, simplified exponential equation. E to the power of negative S equals 0.01. We are now ready to extract the variable from the exponent. Continuing from our clean exponential equation, we must now solve for a variable trapped in an exponent. To do this, we must use logarithms. We take the natural logarithm of both sides of the equation. Taking the natural logarithm of e to the power of negative s simply returns the exponent itself, which is negative s. On the right side, we have the natural logarithm of 0.01, which a scientific calculator tells us is approximately negative 4.6. By canceling the negative signs on both sides, we get our final required sperm concentration of 4.6 units. Notice how incredibly low this number is compared to the 100 units needed in the external tide pool example. This mathematically proves the biological principle that internal fertilization is vastly more efficient, requiring significantly less energy and fewer gametes to achieve the exact same reproductive success. So, we have proven mathematically that internal fertilization requires far fewer gametes. Let us visualize exactly what this looks like biologically. On the screen, you see a comparison of the two strategies. The left panel illustrates external fertilization, showing frogs in an aquatic environment. Notice the massive cloud of tiny dots. This represents the numerous, highly diluted sperm and eggs mixing in the open water. It is a chaotic numbers game. Now look at the right panel, illustrating internal fertilization. Here, we see a cross-section of a confined reproductive tract. Instead of a massive cloud, there are only a few concentrated sperm cells moving directly toward a single protected egg. The diagram perfectly captures what our math just proved. The confined space of the reproductive tract drastically reduces the number of gametes needed, providing a safe and highly efficient environment for reproduction. We have covered a lot of ground today, so let us reinforce the most important ideas from this topic. First, external fertilization relies on producing massive numbers of gametes to overcome the high dilution and dangers of an open aquatic environment. Second, internal fertilization requires more complex anatomical structures, but it provides a safe, confined space that drastically reduces the number of gametes needed. Third, we saw how the mathematical model of gamete collision perfectly proves why internal fertilization is so much more efficient. Ultimately, both external and internal fertilization are brilliant evolutionary solutions to the problem of reproduction, perfectly adapted to where an animal lives. You now have a complete foundational understanding of how life ensures its own continuation. Excellent work today.